This lecture will focus on recombinant DNA technology, cloning, expression, and purification. We'll start off with the basics of DNA. Shown here are the purines, adenine, guanine, pyrimidines, thymine, and cytosine. These are the bases, and when they're attached to the sugar, they are the nucleoside, and when they're attached to a sugar phosphate, they're nucleotide. You should know the difference that in an RNA, this H group becomes an OH group and it's less stable. RNAs are less stable than DNAs. You should also note that adenine, thymine base pair as well as guanine and cytosine, and know how to draw those base pairs. In the structure of DNA, you have it self-assembling. You have two strands self-assembling together to form a double helix. And when they form the double helix, they have essentially 34 angstroms per complete turn, or 10 bases per turn, and then they have a diameter of about 20 angstrom. And as you notice, the outside surface is a phosphodiester bond, and it's negatively charged, and so it will interact with positively charged residues from DNA or other macromolecules. Now, when you have the B-form DNA, as you're shown here, you can see that there are two types of grooves, a major groove, which is slightly larger than the minor groove. Base pairing. Like I said, you will have to know how to draw these bases as nucleotide, nucleosides, and the base pairing. And shown here is the AT base pair, and there's two. And shown here is the GC base pair, and there are three. So this is because there's three hydrogen bonds, you have a slightly more stable base pairing than the AT. And as you can see here, here's the space-filling models of both the AT and GC. And you can see that you have hydrogen bonding as well as the showing of the major and the minor grooves. There is a polarity to DNA, so you have the 5' end, which is the free phosphate, and then the 3' end, which is the free hydroxyl. And so the polarity goes from 5' to 3, where you have the phosphodiester bond on one side and then your bases showing on the other side. When RNA is synthesized, it's synthesized by the addition of new nucleotides to the 3' hydroxy end of a growing chain. And so shown here is the template DNA strand, and here's your RNA being synthesized. And as you can see, it adds to the 3' hydroxy. Here's a triphosphate in the presence of the 3' hydroxy, and then you have reaction where you have the phosphates and the hydroxide react, releasing a diphosphate, shown here, and then forming a phosphodiester bond. The basic central dogma of biology is that you have DNA making RNA, which makes a protein, and so there's a two-step decoding process where you have transcription, making the messenger RNA, which is then translated into the polypeptide chain. Again, here's a flow of the central dogma of biology. Again, DNA makes RNA, makes protein. Here it is in showing that you have DNA being transcribed into rRNA, messenger RNA, and transfer RNA. In the presence of the ribosome being able to be translated into protein. Now, in the genetic code as shown here, there are 64 codons, and the 64 codons encode for 20 amino acids, shown here. You have methionine, which is usually the start codon, and it's encoded by one codon. And then you have 3 stop codons, UAA, UAG, and UGA stop codons. And as you can see, there's a degeneracy in the codons where some amino acids are encoded by multiple codons. So for example, proline, shown here, is encoded by CCU, CCA, CCC, CCA, and CCG. And so this allows for multiple codons encoding for some amino acids. Now we know the basics of DNA, RNA. And now we're going to go into cloning and the basic components that are involved in cloning. So you need enzymes. Particular enzymes that you will need are polymerases, restriction enzymes, and DNA ligase. And then techniques, polymerase chain reaction or PCR, and gel electrophoresis. So polymerase. Polymerase is important for transcription. Structures, so this is an example of RNA polymerase 2, Pol2, which will recognize a promoter region, which is upstream of where you're going to do the transcription. And so you have transcription factors and polymerases work together and help bring this complex, I guess this RNA polymerase complex or enhanceosome, so that it can direct transcription. In this complexosome, or enhanceosome, there's an important sequence, the TATA binding sequence, which is bound by TBP, which is a TATA binding protein, which then allows for the recruitment of all these other transcription factors and transcriptional activators shown here. The other enzyme that was important are restriction enzymes, and restriction enzymes are essentially your molecular scissors. They cut sequences, palindromes that are observed in sequences, and they make either blunt ends, 5' overhangs, or 3' overhangs. So the overhangs generate what are called sticky ends, as shown here. And the 5' overhang you cut in this fashion, where you have an asymmetric cut, and then you have a 5' overhang when you cut. A 3' overhang you cut in the opposite direction, and now in this case you have two 3' overhangs. And then a blunt end takes a palindromic sequence and cuts it straight in the middle, leaving to two blunt ends. The 5', 3' overhangs generated by enzymes that cut asymmetrically are called sticky ends or cohesive ends because they can stick back together. And then the other important, so we have molecular scissors, now the tape. So the molecular tape is ligase, often used in ligation, a process where you take two fragments together, as shown here. These are sticky end fragments that are complementary that will come and join together. So in DNA ligation, it involves creating a phosphodiester bond between the 3' hydroxy of one nucleotide and a 5' of another. The enzyme that's used is T4 DNA ligase, which comes from T4 bacteriophage. This enzyme will ligase the fragments having overhanging cohesive ends and that are annealed together, and then form that phosphodiester bond. It also will ligate fragments with blunt ends, but in that case they'll need higher concentrations of the enzyme for the blunt end ligation to happen. The other component is gel electrophoresis. Shown here is a gel in an agarose gel in a container where you have a cathode and a anode. And so since DNA is negatively charged through the phosphates on the surface, you can then run a current through so that the DNA then migrates to the positive charge, allowing for separation by length. The DNA, again, is migrated by size, where the lightest fragment migrates first and the heaviest fragment is slower to migrate. And so you can use different types of concentrations of agarose, and depending on that concentration of agarose, you can then start to separate different sizes or fragments. So at a high percentage of agarose, you're able to separate very small fragments, but then as you get to large, like small concentrations of agarose, 0.6, you now are able to separate much larger. A vector is an agent that can carry DNA fragments into a host cell. So a cloning vector essentially is used to reproduce the DNA fragment. If it's used to express a gene, it's called an expression vector. There are several common vectors that are used, plasmid being the most common, lambda, phage, cosmid, and even yeast artificial chromosomes. In a plasmid, as shown here, you have a marker that allows you to track the plasmid. Here's ampicillin resistance marker, origin of replication, as well as a polylinker with restriction sites that allow you to clone in various inserts. And the sizes of plasmids range from kilobytes to 100 kilobytes. And usually in such cases you can clone in small fragments of proteins. Lambda phage are viruses that can infect bacteria. Major advantage of the lambda phage is its high transformation efficiency. It's about 1,000 times more efficient than the plasmid. So in lambda phage, you have a linear phage genome in which there's a replaceable region that can be replaced with your target DNA through recombination. And then this is packaged into your lambda phage, and then the lambda phage in the presence of E. coli can infect E. coli, and then that DNA gets transmitted into the E. coli. The cosmid vector is a combination of the plasmid vector and the COS site, which allows the target DNA to be inserted into the lambda head. Advantages, its high transformation efficiency. The cosmid vector can carry up to 45 kilobases, where plasmids and lambda phage are limited to 25. And so here is a cosmid with your COS site shown here. And this is for packaging into the lambda phage. And so here's your polylinker again, where you can add in your gene, and origin of replication, and then some marker that you can track. And so you cleave the polylinker, you ligate onto your target gene, and then you ligate, make a linear cosmid that can be used. The yeast artificial chromosome, or YAC vector, can carry up to 2 megabase pairs of DNA fragments. It has a lower efficiency transfer efficiencies. These vectors contain sequences to replicate and maintain the chromosome in the budding yeast, and contain a yeast origin of replication, in addition to a centromere and telomeres at each end, and of course a marker so that you can track where it's going. So you can take a YAC vector, your target DNA, you digest it with, in this case, EcoR1 and BAMH1. These linearize your YAC vector. You digest your target DNA with EcoR1, and then you ligate, forming this linear yeast artificial chromosome bearing your insert of DNA. Now, when we look at, after you've generated your actual recombinant DNA, you can then amplify that DNA. And so let's look at the plasmid. In the case of the plasmid, where you have a DNA fragment, your cleaved plasmid, then you're ligating with your molecular tape, your insert to your plasmid, and then you take a bacterium, you transform this DNA into your bacterium, and so now that you have your bacterium, it has its host chromosome, but it also has your plasmid, and you can identify your plasmid or track it by using, for example, ampicillin if it has a beta-lactamase gene marker. And then you can specifically grow these isolated bacteria so that you can make multiple copies. Cloning strategy consists of the following steps. You have a gene insert that is amplified by PCR. The PCR product is cloned into a specific cloning or expression vector by digestion with restriction enzymes, your molecular scissors, and then ligation into a restriction vector. The sequence of the positive clone is checked by analysis. So here's PCR. You take your DNA, and you use 2 primers that can anneal to sites of that DNA where it can then amplify the region of DNA that you want. And so these will hybridize onto that DNA, and then through polymerase, it will extend so that you make a copy of your first strand, and then you can do this iteratively where you have a second strand or a second cycle of after elongation and a third cycle. And so in each cycle, the amount of target DNA is doubled. So after 10, 20, and 30 cycles, there's about 1,000 million and a billion fold amplification. So let's look at the PCR cycle. It has 3 steps. The first step is denaturation. So you have your DNA to be amplified as well as your primers. You denature, and then you lower the temperature, and then you start annealing so that the primers can add on to the DNA. And after the annealing step happens, you have extension so that you have polymerase extending out with the dNTPs. And then this undergoes a second cycle and then multiple cycles. So there are some rules for primer sequences, especially at the 3' end of the primer. It's critical for specificity and sensitivity for PCR, so it's recommended not to have 3 or more GC base pairs at this position. This will stabilize non-specific annealing of the primer. A 3' thymidine because it's prone to mispriming. And primer pairs should be checked for complementarity at the 3' ends because this leads to primer dimer formation. Bases at the 5' end of the primer are less critical for primer annealing, so you can add sequence elements at the 5' end of primer molecules, and the length should be about 18 to 30 base pairs long. Shorter primers could lead to amplification of non-specific PCR products. So the TM, or the melting temperature, is important because the specificity of PCR is dependent on this melting temperature. And usually the good results that are obtained have TMs that are above 60 degrees. And the TM for a primer can be estimated using the following formula shown here, where you have 2 degrees times the number of AT plus 4 degrees times the number of GC content. And the GC content of a primer should be at least 40 to 60%. So if you're designing primers, there are 2 types of primers. There's the 5' end of a primer. And so when you're designing the 5' end of the primer, this overlaps with the 5' end of the gene of interest and should contain the following elements: a restriction site, so that'll allow you to clone in the insert; a 5' extension to the restriction site, so that allows the restriction enzyme to actually sit on the site and cleave the restriction site. So usually you want to have an extension that's about 2 to 10 base pairs long. And then the start codon, and the start codon is usually the ATG methionine. This usually should be present. And then after the ATG, the overlap with the gene of interest. So the primer and the gene should be long enough, that overlap region should be long enough to give a TM of 60 degrees or more. For the 3' end of the primer, this overlaps with the DNA strand complementary to the 3' end of the gene interest and should contain the following elements: a restriction site, again, that's the other site, and should be different from the restriction site on the 5' end so that you can have a proper directionality. 5' extension to the restriction site, again, because restriction enzymes need some bases to sit on top of the actual restriction site. You can have a 5' extension that's about 2 to 10 base pairs. And then stop codon. So in preference of which codons, because there are 3 types of codons, the TAA is preferred because it's less prone to read-through than TAG or TGA. And then this stop codon should be included when there's no C-terminal tag used. Sometimes you can add double the stop codons to make sure that it ends. And then overlap with the strand complement to the 3' end of the gene interest. And again, the overlap should be long enough to give a TM of 60 or more. So let's look at this example. Here's a gene, the COAD gene from E. coli. This encodes for phosphopentathione adenyl L-transferase, or a PPAT, an enzyme involved in the biosynthesis of coenzyme A. And so here's a gene sequence, and you're asked to clone this into the PET25B, which has a his tag, using the restriction enzymes NCO1, which is at the 5' end, and BAMH1, which is at the... For the 5' end primer, the NICO1 site in the vector is in frame with the N-terminal his tag, and can be used directly, providing the ATG in the site. So here's the NICO1 site, CCATG, so that it's in, the ATG is in the start of that site, and then everything else should be as complementary as it can be to the PPAT sequence. And then to the 5', there's a 5' extension, again, to allow for the enzyme to sit on the sequence and cut. NICO1 restriction having a start codon, and then the rest being overlapping. When you don't have a situation where the restriction site is has a start codon that's in frame, best thing to do is start the start codon right after the restriction site, and then make sure everything else is complementary. So this is the primer design shown here, and so the TM should be calculating at the start of the ATG. So consider there's 13 ATs times 2, and then there's 10 GCs times 4. This gives you a melting temperature of 66, which is greater than 60, which is good. And then the GC contact for the entire sequence is 43%, and again, the GC contact should be between 40 and 60%. So in this case, the COAD gene has a sequence right after the ATG to be CAA, but because the restriction site has to have a G, it gets converted into GAA, and so the second residue then becomes mutated into glutamate instead of glutamine. And if you want to avoid this, where you're trying to have a sequence where it's the exact same sequence instead of making a mutation, again, like I said, put your restriction site, and then right afterwards add the ATG, and then whatever the complementary. The 3' end primer, in this case, is designed shown here. There's no C-terminal tag, and as a result, we can add a stop codon, at least the complement to the stop codon for the PPAT gene. And so again, you have a 5' extension. This time your restriction site, in this case it's BAMH1, stop codon, right? And then the reverse complement of the 3' M of the COAD gene. Estimated TM, again, you're going to be looking at this region is 60%, and then the GC content for the entire sequence is 60%. So the vector needs to be prepared, and you will have to digest the vector, preferably using 2 different restriction sites. You need to dephosphorylate the ends, and that can be done by adding cafintestin or shrimp alkaline phosphatase. This prevents the self-ligation. Then purification of the digested vector, you can run an agarose gel so that you can remove just Nic and sulfocoiled vector versus the properly digested DNA, reducing the background ligation of the parent. And then the insert. The insert also needs to be digested, so once you've done a PCR reaction, you want to digest the insert with 2 different restriction sites. You purify that again by agarose gel electrophoresis, and then you clone or you ligate the insert with your vector. The ligation will help take your vector and your insert and forms a phosphodiester bond between the adjacent 5' phosphate and the 3' hydroxyl ends. You can use T4, bacteriophage T4 ligase, because it's actually very powerful and has been used to join blunt-ended ends. The efficiency of the ligation depends on the absolute DNA concentration, so you want to make sure it's high enough for that intermolecular ligation is favored over self-ligation. The ratio between the vector and insert, maximal yield of the right recombinant, is usually obtained using a molar ratio vector of DNA insert to vector of approximately 2. But in general, we set up reactions of 1-to-1, 2-to-1, and 3-to-1. Cloning strategy, higher yields of the right recombinants are obtained when the vector and insert have been prepared using 2 restriction enzymes and the digested vector has been gel purified. So once you've done your cloning, you take the DNA, amplify it, extract it, and then use that DNA into sequencing. So in general, a regular base has a 3' hydroxy, which allows for the phosphodiester bond to happen. But for using sequencing, they use this dideoxyribonucleotide triphosphate, in which instead of an OH, you have an H, which prevents it from forming a phosphodiester bond. And so what you do is you have amounts of dideoxy G, A, T, and C, and then you add that to a PCR reaction, and then you run it on a gel, and then based on the length and the color of the product, you can identify what the sequence is. DNA sequencing machines use fluorescent dideoxynucleotides, and so here's a sequencing trace in which you have different base being identified. So each dideoxynucleotide has a different fluorescent tag, so the color of each peak identifies the dideoxynucleotide terminator incorporated at the site. So this is what it shows, and then you can determine. There are different hosts of which you can express your protein, E. coli being the most common. It's the easiest, quickest, and cheapest method. They have different commercial and non-commercial expression vectors with different NC terminals tags and different strains. Yeast is a eukaryotic organism and has some advantages and disadvantages over E. coli. Major advantage of E. coli is that it can be grown to very high densities, which makes them useful for production of isotope-labeled protein for NMR. And the 2 strains that are used are Saccharomyces cerevisiae and Pichia pastores. Baculovirus-infected cells. Insect cells are higher eukaryotic system than yeast and able to carry out more complex post-translational modifications than the other 2. They also have the best machinery for folding of mammalian proteins and therefore give you the best chance of obtaining soluble protein when you want to express a protein of mammalian origin. The disadvantage is insect cells are higher cost and longer duration before you get protein. So there are the different expression systems, yeast, E. coli, insect cells, mammalian cells. They have different cell growth, E. coli being fastest, mammalian slow, complexity of growth. E. coli and yeast is minimal, complexity is much larger. Growth of growth of medium, it's low, high, and high in the cases of mammalian cells. High rates are found in expression levels. It's low to high in insects, and low to moderate. It secretes to the periplasmin. These all secrete to the medium. Refolding is usually required. In the case of E. coli and yeast, that they fold properly. There's no post-translational modification. There is one type of post-translational modification, which is high mannose. You can have simple, no sialic acid, and you have complexes. Again, in the case of E. coli, you don't get any of these, and then the rest you can, with the exception of gamma carboxylation. It can't be happening under high mannose conditions or static acid. So you need to transform the DNA into the E. coli, and in order to do so, you have to make your E. coli competent. For high transformation efficiency, you usually use electroporation or electroporation competent cells. The choice of E. coli depends on the goal of transformation. Usually you're done the transformation in a Rec-A minus cloning strain, such as DH5 alpha, Nova Blue, XL1 Blue. And then you should pick about 3 to 12 colonies and then test by restriction analysis or colony PCR to see if the sequences are correct. The transformation of a vector for multiplication should be done also in a Rec A- strain, such as these shown here. And here are different expression hosts and vectors shown here. So here's a pBAD, PET, PMAL. Some of you have seen this before, and you'll see this again in many of the papers, and then here are the appropriate host strains. So there are different expression systems, as we've discussed. You have E. coli, but there's Lactococcus lacti, Bacillus, and yeast. We discussed Pichia pastoris and Saccharomyces cerevisiae. You also have Pichia methanolica. And insect cells, you have baculovirus, stable recombinant cell line, Schneider cells. Mammalian cells, you have viral infection, usually using adenovirus, semi-liquid forest virus, inducible expression. In vitro is rabbit reticulocytes or red blood cells or wheat germ extract or even E. coli extract. And then there's xenopus oocytes, etc. shown here. Expression methods. So when you're ready to express, you first pick a single colony from a freshly streaked plate, and then you can then pick that colony and put it into liquid culture and then use that as a starter culture to inoculate some larger 50 ml volume or even higher media containing the appropriate antibiotic. When heterologous protein is toxic for cells, higher expression levels are obtained by using the plating method. In the plating method, a single colony of plasmid containing cells is suspended in 200 ml water and vigorously shaken. Suspension is plated on an LB plate containing appropriate antibiotics. All colonies are scraped off and suspended in LB media. The suspension usually has an OD of approximately 0.4. The culture can be grown to the desired OD to start the expression. And once you, in the case of where you just pick a colony and make a starter culture and then start another larger volume, you want to grow them for 4 to 8 hours at 37 degrees. Induction. So once you have your genes in there, you can induce protein expression. Protein expression is induced by the addition of proper inducer or changing the growth conditions. So here are different types of promoters. The TRIC hybrid promoter, you can add IPTG and that'll induce protein expression. Arabad arabinose PL, shifting the temperature from 37 to 42 degrees, and lacoperon by addition of IPTG. Here are incubation temperatures and how much time you need depending on the temperature. There are codons that are not frequently used in E. coli, yeast, and Drosophila and primates. And here are the 8 least codons used in these organisms. And so in this case, what you want to do is when you're identifying a sequence to clone in, you want to make sure that you avoid the ones that are least used. Here's just a table of the 6 least used E. coli codons by selected other organisms, as shown here. Shown in red are codon frequencies of more than 15 per 1,000 codons. And you can see arginines and over here across the different organisms. Protein expression levels ought to be optimized. You can vary induction conditions by changing the time or the temperature of induction as well as the concentration of the inducer. You can examine the codon usage of heterologous proteins. Right? Not all the 61 mRNA codons are used equally. There are major codons that are highly expressed proteins, whereas minor or rare codons tend to be in genes expressed at low levels. Which of the codons that are rare depends strongly on the organisms. Usually the frequency of codon usage reflects the abundance of their cognate tRNAs. Therefore, when the codon usage of your target proteins differs significantly from the average codon usage of the expression host, this can cause problems during expressions. Following problems are often encountered: decreased mRNA stability, premature termination of transcription or translation, frame shifts, deletions or misincorporations, and inhibition of protein synthesis and cell growth. Especially in cases where rare codons are present at the 5' end of the mRNA or in clusters, expression levels are low and truncated proteins are found. The culture can be incubated at 37 degrees until the OD is approximately 1. And then you collect the cells by centrifugation and resuspend them in fresh media and use this to inoculate the main culture. You use, if you're using ampicillin for beta-lactamase gene within the plasmid or your recombinant DNA, the selectable marker beta-lactamase is secreted into the medium where it hydrolyzes all the ampicillin. This point is already reached when the culture is barely turbid. So from here on, cells that lack the plasmid will not be killed and could overgrow the culture, so you need to be careful. Here's some possible solutions. You grow overnight cultures at 30 degrees or lower, spin overnight cultures and resuspend pellets in fresh media to remove the beta-lactamase. Use more stable carbenicillin instead of ampicillin. You can inoculate the main culture and incubate until the OD reaches 0.4 to 1. The optimal OD value depends on the culture method at the media and the medium. And if you're using LB medium, OD of 6 is recommended. To increase growth rate, you carry out cultures at 37 until the OD for induction is reached. Then cultures are cooled to the induction temperature in iced water. For good aeration, just do not use more medium than 20% of the total flask volume. Properly folded proteins are quite stable. Nonetheless, there are some determinants of protein instability that have been elucidated. There's the NN rule, in which the E. coli N-terminal arginine, lysine, leucine, phenylalanine, tyrosine, or tryptophan residues greatly decrease the half-life of the proteins. So a test proteins with these residues in N-terminal position showed half-lives of only 2 minutes compared to more than 10 hours with all the other amino acids. Similar to the NN rule pathway, similar NN rule pathways are present in all organisms, including yeast mammalian cells. And then you want to use the pest hypothesis. In eukaryotic proteins, the proteins are destabilized by regions enriched in proline, glutamic acid, serine, and threonine. Sometimes the level of protein expressions are low because high level of proteolytic degradation of the target protein. So you can use protease deficient host stains, you can do periplasmic expression, or you can decrease the growth temperature to help as well. Site-directed mutagenesis, this is a technique to make mutations. Here you take a primer that has actually the mutation in the sequence, right in the center of the primers. You then denature the DNA template, anneal the mutagenic primers, and then extend the primers and incorporate the nucleotides so that you generate a new double-stranded DNA. Then once you've done this, you can then take you still have your parent plasmid, and you can take that mixture that bears your parent plasmid but subject it to DPN1 digestion. It will only digest methylated DNA, which is where the parent has, and then you can transform your DNA. So to summarize, primer design rules: primers should be at least 15 base pairs long. They have at least 40% GC content. For, primers that are used for site-directed mutagenesis, you want that to be higher, about 50%. Anneal at a temperature range 50 to 65 degrees. Usually higher annealing temperatures are better. Forward and reverse primers should anneal at the same temperature. Primer problems. Primers should flank the sequence of interest. Primer sequences should be unique. Primers that match multiple sequences will be given multiple products. Repeated sequences can be amplified, but only if unique flanking regions can be found where primers can bind. Primers can have self-annealing regions within each primer, like a hairpin or foldback loops, and primers of primers can anneal to each other to form dreaded primer dimers, and you want to avoid them. Typical PCR experiment is carried out under the following conditions. You have polymerase, buffer, primer, DNTPs, polymerase, and your DNA template. 100 to 25 nanograms for mammalian genomic DNA and 20 nanograms for linear plasmid per 50 ml reaction mixture. You want to use a cycle of denaturation at 94 to 92 degrees for 3 to 5 minutes. Then you have amplification, which has this cycle of denaturation, 10 to 30 seconds at 92 to 94, annealing 10 to 60 seconds, 50 to 68, depending on the polymerase, and then extension 60 seconds times the product length in kilobases at 72. And then you can do this from 25 to 40 cycles, and then you have a final extension of 5 to 10 minutes at 70 degrees. So once you have the proteins generated, there are other things that you will need to know is protein purification. You're going to lyse cells, usually either by freeze-thaw, sonication, enzymatic lysis, or denaturants. You can use column chromatography, gel filtration, anion, cation exchange, affinity chromatography, as well as reverse phase chromatography. And then you can also use fusion proteins like GST or his tag. In protein purification, you have a stationary phase, and that's either a gel, resin, or adsorbent, and then a liquid mobile phase, which contains a solvent or buffer. You then use protein sample, and you dissolve it in the mobile phase, and then layer it on top of the column, as you're showing here. The sample mixture enters the column and then is allowed to percolate through the stationary phase as you're pumping more mobile phase through. You have the solutes move down the column, and so then you can separate the individual components in the. For proteins bearing either tags like GST or his tags, in the case of GST tags, you can use glutathione. So you have, here's a magnet glutathione particle of which GST tags fuse to the protein, bind to. You can wash away the non-GST containing proteins and then elute out your protein of interest. In the case of his tag proteins, which you'll see in many of the papers, you can do it under native conditions or under urea. In the case of native conditions, you're going to bind and then elute out based on concentration of imidazole. So in this case, you have 100 to 250 imidazole purifying out the protein. In the case of denaturing conditions, you're using urea or guanidinium hydrochloride. And then in this case, you're using pH. So as you lower the pH, so you can use this at pH 6 to wash, and then at pH 5.9 or 4.5, you start to elute out your protein. So this concludes this lecture.